Good morning, everyone. Thank you for joining us at our webinar Beyond MOSH and MOAH Analysis presented by Neotron. Technique, Risk Evaluation, and GC GC-TOFMS Confirmation. We start with the speaker presentation. I'm Barbara Moretti, and I'm your host for today's session. I'm the head of the packaging department, the Neutron division where the mineral oil analysis is in charge. I have a chemistry degree and I've been working in Neutron since 1999. I will give you a brief introduction on mineral oil topics. Next up is Valeria Mazzoni. She will introduce in techniques and risk assessment evaluation. She has she has industrial technology degree, has been working in Neutron since 2017. She's analytical R&D and risk assessment specialist. Finally, the floor will be for Erika Vellati. She will introduce in GC GC-TOFMS FID confirmation. She has a degree in chemistry. She has been working in Neutron since 2007. She's a senior GC analytical specialist with focus on GC by GC technique. This is our agenda, and we tell about a brief corporate and Neutron overview, an introduction to MOSH and MOAH analysis, technique and risk evaluation, GC by GC-TOFMS FID confirmation, and refer to the Q&A section at the end of our webinar. We received many questions during the registration process, and we selected the most frequent ones. But if you have any question in the middle of our presentation, feel free to use the Q&A section or chat box. We will address those questions toward the end of our program. To give you a brief introduction about our company, Cotecna is a Swiss-based company headquartered in Geneva, with offices and labs across the globe. It is a leading provider of testing, inspection, and certification services. Today, now have over than 7,400 employees, with more than 180 offices and laboratories. Our trusted network of professionals and certified laboratories provides expertise across our 5 key sectors, namely agriculture, food safety, metals and minerals, consumer products, and system certification and ESG solutions. Neutron is the biggest laboratory of Cotecna Group. Located in Modena, in the north of Italy, more than 550 employees work in 10,000 square meters of laboratory by managing more than 1,000 samples per day and more than 5,000 accredited tests. These are the field of the testing activity, which Neutron has activated during its 50 years of history in relation to the market and in relation to the customer requests. Now, let's start. Mineral oil hydrocarbons gather several mixtures of hydrocarbons containing thousands of chemical compounds of different structure and size. Mineral oil are easily composed of 2 chemical and structurally discrete types of fraction, MOSH and MOAH. MOSH, mineral oil saturated hydrocarbons, that consist of a complex mixture of saturated hydrocarbons with open chain or naphthenic structure. Within this element, MOSH analogous must also be considered. These synthetic hydrocarbon substances group highlighted are particularly relevant. For example, polyolefin saturated hydrocarbons are oligomers resulting from the polyolefin polymerization process. Polyalpha olefins are synthetic lubricant or hot melt adhesives. Mineral oil refined products include all the other mineral oil refined, such as paraffin waxes. To their chemical similarity, MOSH analogues are very difficult to separate from the MOSH fraction using conventional method. the present cause overestimation. I said conventional method because there is a different method that allows to separate them, but I don't want to anticipate anything because it will be explored in depth during the webinar. The secondary fraction is composed of MOAH, mineral oil aromatic hydrocarbons, a mixture of highlight alkylated aromatics with the last one aromatic ring system. These molecular properties increase their chemical reactivity. In fact, MOAH fraction brings a higher concern relative to the MOSH fraction. In general, the ratio between MOSH and MOAH is 4:1. The evaluation of this ratio can be used to indicate a mineral oil contamination. This is our history from the past to the future. More than 13 years of experience in mineral oil analysis. We started with offline separation, and several equipment have been acquired during the year, but not also. We are working in several European working groups for the method optimization to demonstrate our commitment to ongoing scientific research and innovation. For this year, we are waiting the new Access MRAW equipment. This instrument will be equipped with the MOSH depletion. New tools useful to manage the high concentration in saturated hydrocarbons, like in sample, cosmetic sample, for example. Mineral oil contamination has been talked about for many years, but the issue is set to take a hot topic for a long time in food safety. As can be seen from the notification posted in the European Commission RASP window, since the beginning of the year, the borders control are continuing and several matrices are involved, like tropical oil, cocoa, and new one like thickener, such as guargam. It is increasingly the importance to identify the source of contamination in order to eliminate it. All entire food chain is involved. Neotron is every day in first line for the food safety, and today we are talking about the importance of analytical tests, the risk evaluation mandatory to identify and manage the sources of contamination, and GC FID test as essential tool that help in the mineral oil investigation. Before leaving the floor to Valeria Mazzoni for a specific in-depth presentation referred to the analytical technique and risk evaluation, I remember you to report in chat your question. Now let's move to our second speaker, Valeria Mazzoni. Valeria, please, the floor is yours.
Good morning, everyone, and thank you, Barbara, for this great introduction, and thank you, audience, for joining us today at this webinar. My name is Valeria Mazzoni, and I'm here to explore with you the implication of mineral oil contamination, particularly focusing on analytical techniques and risk management strategies. Mineral oil, primarily in the form of MOSH and MOAH, can inadvertently find its way into the food supply chain through various sources, and understanding and managing these contaminants is a critical point today. In this first part of the presentation, I will first outline the pathways through which mineral oil contamination occurs. Then we will delve into the analytical technique currently employed to detect and quantify MOSH and MOAH in food products. And finally, I will discuss risk management strategies that can be implemented at various stages of the food supply chain to minimize the presence of these contaminants. And by combining effective analytical technique with the proactive management practices, we can better safeguard our food supply and public health. So let's get started. Understanding the sources of MOSH and MOAH contamination is essential for developing effective prevention strategies. Today, the complexity of the issue is well known, and addressing the contamination pathway is becoming more and more challenging. And as you can see, the consideration of this topic has been expanded to different sources of contamination. It now includes all possible routes of entry along the entire production chain for food and food packaging. So you can see that we have to consider farming, starting from farming, going down to processing and storage, considering also all the intermediate packaging, and of course the final primary packaging or secondary packaging, like recycled cardboard or plastic and so on. But we must also consider environmental effects and transports like emission from energy supply chain or exhaust fumes from combustion engines. But as we can see, all these kinds of contamination have a typical chromatogram profile that generally takes the form of an AMP. And the peculiarity of mineral oil analysis is that on the basis of the distinct chromatograms, we can hypothesize the nature of the contamination. This key of interpretation can give a strong contribution to the risk assessment in the food and food supplement production chain. However, we must face with a high complexity of the analysis that leads to the differentiation between the MOSH risk evaluation and the MOAH risk evaluation. In fact, according to the latest EFSA opinion of 2023, MOSH fraction is not of human health concern, but bioaccumulation is possible. So a margin for safe exposure limit must be considered, and in case of high MOSH values, mitigation measures has to be provided. In the MOAH risk assessment, two different evaluations have to be undertaken. One for the 1-2 aromatic ring part, for which, unfortunately, insufficient toxicological data are available, and one for the 3 up to 7 aromatic ring that, on the contrary, are strictly under the crosshair due to their genotoxic and carcinogenic nature. As this issue continues to evolve, ongoing research and regulatory efforts will be vital in mitigating the risk associated with these contaminants. And the European Commission is constantly working on a continuous improvement of the analytical methods to further investigate the source of the contamination and contemporarily collect as much toxicological and also chemical composition data as possible to cover all gaps. And in order to achieve this goal, in this month, the European Commission is working on a regulatory follow-up of the EFSA opinion. And in fact, a draft regulation of the maximum limit of MOAH is under discussion, and hopefully next year it will be released. In this proposed regulation, the MOAH maximum limits are currently similar to those already identified in the April 21 joint statement of the member states, for which the products can be distinguished into 3 macro categories, as you can see. So products with fatty content below the 4%, product with a fat content that is between 4 and 50%, a product with a high content up to 50%. And it is specified that the levels are considered applicable for both business-to-business products and business-to-consumer products. Moreover, in order to ensure a uniform enforcement approach throughout Europe, the member states agreed to withdraw and, if necessary, to recall products from the market when the sum of the concentration of the MOAH in food are at or above the maximum LOQs. In the context of the EFSA opinion on mineral oil contamination, the role of analytical methods in ensuring food safety cannot be overstated. And it's by providing reliable data and insights that regulatory compliance, risk assessment, and effective risk management strategies could be really assessed. So let's focus on the general analytical method currently in use. MOSH analysis has been designed specifically for sensitive and fast detection of mineral oil contaminations by the JRC guideline of 2023 and consists of a sequence of procedures that conducts to the final quantification of the contamination. Of course, every food matrix has its own path, but in general, we can resume it with the following approach. So after a first homogenization, the sample is dissolved in hexane and start the saponification in order to remove the lipidic part and enrich the sample to improve the analytical sensitivity. Subsequently, we perform a liquid-liquid extraction with hexane to remove as matrix components as possible from the sample. So we proceed to one of the main critical steps of the process, that is to say the epoxidation step. This step can be essential for the removal of the MOAH interferences, generally represented by the olefins, so natural polyunsaturates monoterpenes like carotenes, squalene, or stearins, that can coalute during the HPLC separation. And as we can see, they show broad peaks in the chromatogram that can lead to an overestimation of the MOAH fraction. MOSH fraction also can present natural interferences, and in this case, the strategy of an additional cleanup technique with the use of an aluminum oxide column becomes necessary for the removal of odd-number biogenic alkanes and natural paraffins. The determination of the true fraction is achieved by applying online coupled HPLC-GC FID system that gives a complete separation and quantification of the MOSH and MOAH fractions. As said before, this is a generic approach, but the huge variety of the matrices can generate deviation from this main path, leading to a case-by-case approach. Using standardized methods for the analysis of MOSH and MOAH ensures consistency, accuracy, and reliability of results across different laboratories. This can be crucial for regulatory compliance, especially in sensitive products like infant formula and oils. For this reason, analytical methods need to be supported by established standards from organizations such as DG Santé, JRC, and ISO, playing a vital role in monitoring MOSH and MOAH contamination. So, focusing on infant formula, here, I show you a summary of the extraction method validated through an interlaboratory comparison attended by 26 laboratories all over the world, between whom Neotron. While for vegetable oils, an international standard, ISO 20122 of 2024, provides guidelines for the determination of the MOSH and MOAH with a specific extraction method for low limit of quantification. And here again, a summary of the main step of this complex method for which Neutron participated to the final drafting. Ensure reliability, accuracy, and robustness of analytical data is fundamental. For this reason, the urgency of fully validate analytical methods must be highlighted. In the draft version of the Regulation 333 of 2027, some criteria will be fixed, such as you can see, recovery and intermediate precision. And for this reason, it is recommended to validate sensitive analytical methods in order to achieve the target LQs for both MOSH and MOAH, and MOAH in particular. And it will be necessary also to validate analytical methods on two-dimensional GC technique in order to allow to distinguish MOSH and MOAH contamination from all naturally occurring interferences. But you know better than I do that this kind of analysis is anything but simple, and unexpected values are always just around the corner. Therefore, systemic process to identify, evaluate, and manage risk associated with potential hazards become crucial. So let's focus on the main step of the risk assessment. First, risk identification that can comprehend the source assessment with the identification of all potential sources of contamination across, for example, processing environments like equipped lubricants or machinery cleaning solutions, raw materials, and packaging materials, of course, too, like recycled paper, plastic, and storage packaging. In this context, vulnerability assessment must be evaluated in order to assess which type of food products are most at risk, considering factors such as fat content or processing methods used and storage conditions too. The second step is the quantification of the contamination. Of course, in this field, advanced methods for quantification as GC FID must be taken into account. And when the limits of this technique are too impactful, other in-depth tests are needed. So GC by GC technique can unravel the skin for the analysis of complex mixtures, giving as outcome both qualitative and quantitative data. Finally, I would highlight the importance of risk mitigation strategies. And for this matter, three main points has to be considered. First of all, control measures in order to implement controls at identified contamination points across the supply chain. For example, ensuring that packaging materials meets safety standards. or maintaining proper protocols for processing equipments. Then risk mitigation strategies can include regular monitoring programs for MOSH and MOAH levels, as routine testing during production, and random sampling in retail environmental. Moreover, collaboration with stakeholders will be recommended in order to share information and best practices for managing the mineral oil contamination by working with suppliers, regulatory agencies, and industry groups. But let's look at real cases to better contextualize the risk assessment approach. Here you can see an example of mineral oil contamination in vitamin soft gel capsules. This case study is as peculiar as representative. In fact, here you can see that we have a clear MOSH contamination with a quite high value. And based on our previous consideration about the increasing importance of mitigation strategies, in this case, a posteriori risk assessment approach was proposed to the client. So, starting from our final product, we decided to break it down by its raw material and perform the mineral oil analysis in order to identify the route of entry of this contamination and concurrently characterize its nature. And considering the similarities of the chromatograms and the presence of two humps, we can conclude that the capsules have been contaminated by both paraffin and mixed oil solution. Here, another case study I would like to show you. In this case, we are in presence of a contamination of gummy product in MOSH fraction. And as shown before, we started from the risk identification, working in a straight collaboration with the customer in order to establish a classification of the major potential sources of contamination across the production chain. So after this initial evaluation step, a gear lubricant used in the final filling step of the product was selected as a highest risk component and analyzed. But as you can see, from the comparison of the two chromatograms, the final product and the lubricant, the first hypothesis of the contamination wasn't confirmed, leading to a necessary deepening of the risk identification process. And if you are a fan of Murphy's Law, you will know for sure that nothing is as easy as it looks. In fact, most analyzed samples are not easy to evaluate, and together the complexity of the matrices coupled with the limitation of the analytical technique, both in an extraction step and non-specificity of the FID detection, necessarily leads to a stalemate. So what can we do in these cases? Starting point could be working on the optimization of the extraction methods with a matrix-based approach. As said before, the high variety of food matrices must be considered and handled case by case. Then, a qualitative confirmation of the real presence of the contamination would be a further step to have a clarification and also a definition of an eventual overestimation of the quantification data given by the presence of eventual interferences. But the real goal is to couple confirmation and quantification of the hypothesized contamination in a unique step given by the GC GC-TOFMS FID technique. And for a deeper insight into this complex topic, I leave the floor to Erika.
Thank you very much, Valeria, for your presentation. And now let's move for the final topic with our third speaker, Erika Vellati. Erika, please, the floor is yours.
Thanks, Barbara, for the nice introduction, and really thanks everyone for the great interest in this topic. My name is Erika Vellati, and today I'll be your guide to a deep journey into MOSH-MOAH universe in order to answer to two questions: what's inside the hump, and finally, how much is it? So let's start. Our journey starts 5 years ago, when we performed our first installation of a GC GC-TOFMS Pegasus from LICO. GC GC is an acronym for Comprehensive Gas Chromatography and represents the most recent evolution of chromatography in presence of complex mixtures like MOSH and MOAH, that can be resolved by simple GC. Basically, it starts from the idea of putting two different separation systems together, initially only for a selected window of time, just like the ERCAT technology, and then for all the GC run, and from here the term's comprehensive. Since columns are opposite, we talk about orthogonality. In order to maintain the first column separation and not to go back to chaos by directly installing the second column, we need something between these two columns. It is called a modulator. The modulator is a device that is able to stop, merge and focus and inject our analyze into the second column in a very, very short period of time. We are talking about seconds, allowing the second column, which is generally shorter than the first, to separate analyze without losing the first order achieved before. For MOSH MOAH in particular, we work in reverse phase configuration. So we have the first dimension, which is a mid-polar phase, and the second dimension, so the second column, which is an apolar phase. The choice of the modulator was a thermal cryogenic modulator, which is a linear dual stage for jets, and the detector is a time of flight, a TOF. This detector is fast enough to collect all the data at the end of the run, and quadruple isn't so effective. So what was the output? A beautiful dimensional chromatogram, which is called counterplot, in which we have volatility and apolarity as axes, spreading all over C10 to C50 and more acquisition range, so perfect for MOSH MOAH analysis, showing us everything that was inside the sample and inside the hump, like a sort of a fingerprinting, and giving us the possibility to classify them with spectral and space filters, but without the possibility of quantifying, not due to the detector use, but due to the particular nature of the amp itself, because we have different composition of analyze in different samples. So we decided to boost our system with a couple of updates. We changed the injector from splitless to MMI on the top line. And we added a second twin line of columns, exactly like the first, equipped with a COC injector, and at the end, of course, an FID detector. We also added an autosampler in order to perform concurrent large volume injection on the two columns line together. Since the thermal modulator, the column separation mechanism, is the same for both lines, and column parameters, just like length, internal diameters, flows, were carefully engineered, we expected two fingerprinting of the sample well aligned. It was so. Yes, it was so. We obtained these two beautifully coupled signals, and all the info that we obtained before, range, classification, separation power, were still there, and a lot of plus were present, like large volume injection, two signals in one run, and the possibility to answer to the question, how much is it, by quantifying only the FID signal. In order to check signal alignment and separation power of our new brand instrument, we started by injecting our MOMIX standard, gently provided by LICO. MOMIX is a mix of all MOSH and MOAH markers from C10 to C50 and over. So we were able to create our full classification composed by different classes. For MOAH, all the aromatic rings, spreading from 1 to 7 rings, including heteroatomic compounds like benzothiophenes, dibenzothiophenes, and naphthobenzothiophenes, and also contamination markers like disopropyl naphthalene that comes from Utah bags and recycled paper. For MOSH, we have everything inside, just like paraffins, can be linear and branch, olefins, naphthalene, which are cyclic hydrocarbons, and all the markers like pristane, phytane, styrane, and opane. These markers are typically of fossil origin, then derived from saturation of biogenic terpenoidic substances from vegetables of the past. The separation power was so big that we can clearly see also the POSH that comes from plastic packaging, just like polypropylene and polyethylene, and it comes from this contamination and also other contamination from virgin and recycled packaging like resins, inks, and dot-mels. So it was time to deal with real samples. But how is the total analytical approach? As seen before, and very briefly, I will go and says that the sample undergoes several steps from preparation, purification, and so on. And finally, we have the injection of the traditional GC FID system as seen before. This approach is very fast, works quite well, but it gives no info on the AMP composition. And there are several cases in which the output isn't so clear. And of course, LOQ must be raised. In this example, we have a MOAH chromatogram where we have too many biogenics interferences over the hump. Here, we have both interferences and the hump, and the total MOAH amount is critically near our LOQ of 1 ppm. Here, the internal standard area is compromised by the presence of interferences, so quantification can be really tricky. Regarding MOSH, sometimes confirmation of polyalpha olefins and POSH from plastic origins is needed. So what we should do now? Okay, we proceed to the next step. The same extract obtained before is now separated in MOSH and MOAH fraction by LC. Fractions are concentrated to a certain volume and are submitted to double and concurrent injection in our brand new instrument. After data collection and data processing, data are analyzed. First, focusing on a mass signal, data are inspected, peaks are identified, and the first classification is drawn with both spectral and space filters. Then, regions in classification are adjusted. If biogenic or other interferences must be removed, and the new classification is then applied to FID signal. Since the two signals are aligned by hardware and software, we are sure to apply the same space filters to this new detection. Last, in order to quantify only the data inside the hump and not above, riding peaks are cut using the smoothing function. Finally, now that we have all the data that has been identified, cleaned by interferences, and trimmed, we can do quantification of total and subclasses of MOSH and MOAH with respect to internal standard. Let's see a few applications. We start with a tropical oil extract, and this is a typical MOAH GC FID chromatogram showing, unfortunately, both hump and interferences. We have quite high level of total MOAH quantified. Interferences might lead to an overestimation of MOAH. A riding peak subtraction is not enough, and we don't have any clue on hump composition, for example, number of rings involved, risk assessment, and so on. So, this is the output of GC by GC. We can easily see that the AMP is now cleaned by biogenic interferences because they fall in different areas of chromatographic space, and that the same fraction injected before presents also other interferences that are not MOAH, but clearly can lead to overestimation of data. Focusing in particular on the MOAH AMP area, we can see that biogenes can also be classified by number of rings and subtracted to the corresponding aromatic classes. This leads us to a total MOAH integrating from 5 to 3.8, mostly divided in monoaromatic and diaromatic MOAH. Application 2, we focus on the MOSH fraction, and we can clearly have identification of other classes like, for example, polyalpha olefin oils that comes from synthetic origin, are polymerized generally from dicene and dodecene, are used as lubricants instead mineral oils in particularly tough condition of temperature and pressure because it works better than the mineral ones. In GC by GC, they are cluster shaped, can be nicely seen also in GC by GC, and must be quantified inside paraffin classes because they are, of course, MOSH, even though they are from a synthetic origin. In second case, we have POSH that comes from packaging contamination, generally from polyethylene or polypropylene, and they can be seen in a different way. In this particular case, we are seeing a polypropylene contamination because POSH are above paraffin's area. And since POSH are not MOSH, they come from packaging contamination and not from a mineral oils one, they must be quantified alone if it's possible. And we can see that GC by GC isn't afraid of this task. In application 3, we switch to a different kind of matrix, from oils to powder, in particular to an emulsifier agent. Submitted a sample to routine GC FID analysis, we found very, very high values of both MOAH and MOSH. Since this matrix is basically without fat and sample preparation is quite simple, we have a liquid-liquid extraction, no purification steps, we suspected a mineral oil contamination in the process or in the raw materials, plus some packaging contamination. POSH from where? To check all of this and to have more info on the composition of these arms, we submitted the same extract to GC by GC-TOFMS FID, and we found out for MOAH. The contamination was confirmed. Many aromatic rings compounds were present, also 3 and more. No biogene except squalene were interfering the result, and the value quantified was the same of the routine analysis. Regarding MOSH, all the markers of the mineral oils were found, and we detected and quantified also some POSH, in this case, from polypropylene packaging contamination. So, we confirm routine analysis output and the mineral oil contamination, probably related to how materials were handled. A few last words to recap all that has been said and before, and some technical limitations. We have seen so far that in MOSH-MOAH analysis, sample preparation and LC fractioning are crucial steps. Unfortunately, matrix and sometimes sample related. One size doesn't fit all. And we need a case-by-case approach with sometimes a long time involved. In particular, MOSH-MOAH separation can be partially incomplete. MOSH can coelute in MOAH fraction as well as in many other interferences. The only way to look inside the true MOAH fractions is by GC. Every manipulation step of the sample, saponification, epoxidation, purification, and so on, can lead to a loss of MOAH and can also add unwanted interferences, over-quantifying the LC-GC output. Purification generally must be applied carefully, and in general, MOSH-MOAH analysis should be tailored on the sample. Luckily for us, GC by GC separation power can correct all of these problems and can easily deal with the unpurified and partially overlapped samples. But, of course, too much is too much. In this example of a coconut oil, sample preparation didn't work well, too many interferences were left, separation was not complete, and too many classes of MOAH were overlapping. In this case, cutting out biogenic areas from the counter plot surely leads to an under-quantification of the MOAH. So, what we should do we need to start from the beginning and define a new sample preparation and eventually a new separation approach, and probably a long time will be involved. So, LC-GC feed technique should be considered as a screening technique because it's very, very fast, can be automatized, work with many, many samples, and is okay for a threshold level of total MOSH and MOAH. And GC by GC TOF FID should be used for confirmation and quantification of specifically classes and subclasses of mineral oil contaminants and a critically total amount of MOSH and MOAH.
Thank you, Erika, for your very interesting and high-level scientific presentation. And thank you also to Valeria for a very interesting and specific presentation. Now we'll open the floor for the Q&A section. We have received a lot of questions, and we have selected the most frequently asked question that you send us during a webinar registration. We start with the first. Is it possible to separate POSH from MOSH and to detect other contamination from recycled plastic packaging? Erika, I think you have just answered to this, this question, but if you want to say more detail about, thank you.
I've said before, separation and quantification of POSH from MOSH is possible only with the GC by GC TOF FID equipment. Plus, in this case, since the configuration is the same, we can also are able to investigate the contamination from plastic and paper packaging by detecting markers of resins, hot melts, phthalates, recycling process contaminants. Of course, depending on the shape and the position of these contaminants, they can be cut out from the total amount. So yes, it's possible.
Yes, it's possible. Thank you. Second one. What happens in case of coelution of compounds whose concentration differs several order of magnitude? I try to answer. The MOSH and MOAH fraction are separated and quantified in the same extract. If the MOSH are present in concentration greater than MOAH fraction in several order of magnitude, the separation may be compromised with overlap of MOSH fraction into the MOAH fraction. At the moment, it is necessary to dilute the sample with an inevitable increase in the limit of quantification. It may be useful, the MOSH depletion, because it's a specific tool that helps to manage the high concentration in saturated hydrocarbons, for example, in sample like cosmetics. Okay, go on. What are contamination sources and how can be avoided in dry food matrices like rice or thickeners such as gum? This is for Valeria. What do you think?
As seen during the presentation and the first part, sources of contaminations are various and can occur during the entire food chain. So we can focus only on packaging materials, for example, that come in contact with the final product. But we must consider all the routes of entry along the entire production chain and all the main characters on this field. So in this case, in dry food matrices like rice and thickeners, sources of mineral oil contamination can be varied, and measures to prevent this contamination must be carefully planned with a risk assessment that would identify the highest risk factors, and of course also which can be the most critical point, for example, in the production chain. So to prevent mineral oil contamination, a combination of appropriate material selection, starting for example, from raw materials and going down to storage, packaging, or storage condition, or also, of course, primary packaging, and combined with a rigorous quality control measures must be implemented to reduce, mitigate, and if possible, of course, avoid this kind of contamination.
Thank you. Very clear, Valeria. Thank you. Go on. It is possible to analyze mixed tocopherols matrix and herbal extract. Very, very challenging answer. Erika, what do you think?
Okay, this is a tough argument, but since I am a chemist, I don't accept a no as, as an answer.
Right,
so, yeah, what can I say? Okay, for herbal, spices, food supplements, matrices is possible, but a step-by-step approach must be taken in account in order to separate MOSH and MOAH efficiently, avoiding interferences. In this case, a deep knowledge of the composition of the matrix is essential, in particular focusing on what we don't to inject on our system, so in particular focusing on interferences. With tocopherols, there are more issues because tocopherols are diterpenes that tend to collude in both fractions. So the MOAH fraction in particular is highly interfered if tocopherols and tocotrienols are present in a bulk concentration. So due to this high amount generally involved, purification steps will be poorly applied and LOQ will be erased generally. In these cases, the search for oil contamination would be better applied to singular raw materials instead of the final product in bulk concentration. But yes, we should work on it, of course.
Okay, and we consider a case-by-case approach because it's very challenging matrices. Last question. Are there any raw material foodstuffs that present particular challenges for MOSH-MOAH analysis? Which ingredients can influence the MOSH-MOAH measurement results? As just explained by Erika, probably the most challenging matrices are spices, herbals, and food supplements. For spices, we are working, for example, in collaboration with the European Spice Agency, with other European laboratory to evaluate criticality about MOSH and MOAH methods. The possibility to having a wide variability within the matrix spice, for example, does not allow an easy standardization of a method because of the magnitude of a natural compound with different analytical behavior, and this is a very big problem for this family of samples. We believe in scientific exchange in international working group and working tables that will allow to goals and to be achieved more quickly. And this is our mission at the moment, to try the best solution for the challenging matrices. For the other question, which ingredients can influence the MOSH-MOAH measurement results? Valeria, could you help me to answer to this question on the basis to your experience?
Yeah, of course. This is quite challenging question, of course, because how many ingredients can be part of the final product and can interact with to the final quantification data. And some ingredients can influence, of course, the final results. For example, as shown before by Erika, the most known interferences is made by tropical oils, for example, that may be found in the final products, for example, bakery or dessert products. And despite tropical oils is only a part of the ingredient and not main one, the chromatograms of the final product reveal its typical shape, and of course we must consider in the final quantification. So in this case, of course, also GC by GC-TOFMS FID technique can be involved in order to quantify the real contamination, the presence of the contamination, and avoid as much as possible the quantification also of the eventual interference.
And thank you, Valeria. We received a very lot quantity of the question in our chat box and the Q&A and answer. We 2 minutes more to try to answer for some of that. For example, one request is referred to the if there are now the legal limit for the MOAH, if the regulation was published, and if there is a legal limit for the MOSH. And we received several questions referred to the legal limit, and the answer is no. And for the MOAH, we are awaiting the publication of the regulation that at the moment is in the draft form for the beginning of the next year, and at the beginning of the 2026. Referred to the MOSH, the legal limit will not be published, but the European Commission are moving in the direction to prepare the reference value. Okay, that are not a legal limit, but can help the food industry to evaluate if the contamination is present in the food. The limit that at the moment are proposed as a recommendation is different in different fatty content, is 5, 10, and 50 milligram per kilo for different quantity of the food, and we don't know if it will be really published, but maybe at the moment a very important threshold for the evaluation of the contamination.
And if I can add a point
yes,
to stress a point for MOSH, I said before during the presentation, the European Commission is stressing the importance also of MOSH, even if there is only not a human health concern, of course, as MOAH, but the stress is on the mitigation point. So we must consider always a mitigation strategies, in particular for when MOSH values are very high, because even if we don't have carcinogenic or genotoxic activity as the MOAH fraction, by accumulation also is possible, and the EFSA is working on this point to evaluate as much as data, toxicological data as possible during these years. So we must focus on this risk mitigation also for the MOSH fraction.
Okay, thank you. One other question that can possible to answer is referred to the contamination of the MOSH MOAH from the PP film. Regarding a primary PP film, which MOSH MOAH threshold should raise concern and lead to suspect of MOAH contamination during film manufacturing. It not exists a threshold, and it not exist a limit of MOSH MOAH in the packaging in general, not in the recycled paper, Utah bags, or other kind of packaging. At the moment, the content of MOSH MOAH is considered like a specific migration. You obtain a concentration in content or in migration, and it's possible to evaluate the possible migration to the food referred to the ratio, real ratio, surface and volume of the specific uses of the packaging. I don't know if I answered to the question, because it's not easy without the knowledge about the quantity, but we suggest to analyze to evaluate the content of the MOSH MOAH, or to perform the migration in food simulants, and to, if the POSH are present, and if you want to evaluate if POSH are present with MOSH, for example, the GC by GC can help to have the complete idea of the situation of migration of the sample. Okay, sorry, we are arrived at the end of our webinar, and unfortunately the time is not enough to answer to all your questions regarding the chat box, but we will answer you by email in the next day. Please, Dana. Okay. We came to the end. We hope you find this webinar beneficial, and moreover, your online participation from so many countries from different continents, meaning that the interest in the mineral oil topic is not anymore an European concern, but all the stakeholders are involved and in food chain. A big thank you to our speakers, Valeria and Erika. Very, very thank you. Thank you for sharing their expertise. Thank you to Dana Sanchez and all the Cotecna team that help us in the organization of this event. And last but not least, thank you for all of you for joining us. We will send you an email in the coming days containing a survey form, a recording of this webinar, and the relative presentations. Feel free to contact us. For the other question and request, you can find in this slide our contact, Cotecna contact and Neotron contact. Thank you. Thank you very much. Have a great day, everyone, and see you in the next webinar. Thank you, and bye-bye.
Thank you.
Thanks.
Thank you.
Thank you.